Thank you. So again, let's go back into protein function, or what protein function is, subcellular localization. This is a eukaryotic cell. There are compartments, different compartments. There are examples for proteins that are valid for specific compartments. This is the ribosome. This is in fact one complex with proteins and DNA and RNA. Collagen is a structural protein for the skin, and here you see examples of membrane proteins here. This gray, hardly visible, but you may still appreciate is in fact a symbol for the lipid bilayer, the membrane, and that is a motor in the membrane, signaling proteins. Subcellular localization is so interesting to predict because, let's begin with something. When I look, the whole pie here is all the drug targets. So drugs are small molecules that target proteins. Target means they are effective against a particular protein. Most of these proteins are in the membrane. Okay, why do you believe this is the case? So why do you believe most of the proteins that are targeted by drugs are in the membrane? So 50% of the pie would be here, right? This blue is larger than 50%. This is the fraction of membrane proteins targeted. 100% is all the proteins targeted by drugs, all the drugs on the market, and over 50% of those are membrane proteins. Why do you believe this could be the case?
Maybe it's easier to target it.
Why?
Because, for example, for the nucleus and so you have to go in, and for the cytoplasm, so you have to go through the membrane, so you have to find.
That's an interesting idea. That's an interesting idea. This is a very yes, actually, it is true. So it is more difficult to enter, but this typically for today's pharmacology or pharmacological experts, that does not pose a threat. So, but you're totally right. It's true. For a long time, this was not that easy. But that barrier, so experts can cross. Okay. Easier. Well, you could imagine that they are sort of trapped in the membrane; they cannot get out, and that could be easier. The real thing is every drug or every reagent that the cell doesn't want. Virus, bacteria, the first line of defense against anything that comes in is the membrane, right? And in fact, even already some proteins that are swimming around outside. And you see immediately here, one of the next bigger compartments is in fact extracellular. So this is in between. So, you come into the cell and the first region here, cytoplasmic extracellular, and you see with these, you essentially have almost all the drug targets. At least a very high fraction of all the drug targets. Now, this again is one of the reasons why, in fact, subcellular localization is an important thing to look at. There are here examples of diseases that, as many other, essentially all the slides today come from Tanya. So what we see here, what I show is a name of a protein. Then I show a disease, or she collected these data. And here's the mechanism, which is in fact, in this particular case, it says mutation of a nuclear localization signal. So this means that the disease is caused by having a variant in that nuclear localization signal. The nuclear localization signal no longer works, the protein is no longer transported to the nucleus, and there's a damage that comes from it, in this particular, the Swire syndrome and other syndromes that are similar. This just lists what alternative subcellular localization you see. Well, loss of nuclear localization typically means often, in fact, it's retained in the cytoplasmic, in the cytoplasm. Outside the nucleus, it just doesn't enter, so it stays outside. Loss of nuclear localization essentially means, I believe, that there is no clear experiment that shows it. What they see is that it doesn't enter the nucleus, and that causes the disease. This is the way to read this table. I guess that the first and the second, and actually all of these are the same. It just stays in the cytoplasm, most likely. But experimental data says we don't know, but we know it's not nuclear, and that causes the disease. Here's just a couple of examples for these diseases that in fact show hit on in all of these cases here, we have something that is essentially attacking the zip code, yeah?
But are those due to viruses or is it
No.
genetic problems?
So these are genetic problems. That has nothing to do with the virus. So these are examples. So again, remember I said, and I will get back to that, between every pair of us in the room, we have 20,000 variants. So 20,000 amino acids differ between any pair of us, okay? That means in every single protein, since we have 20,000 proteins, in every single protein we have one difference. And here are typically, I do not know whether those are in fact point variants, so meaning just one amino acid is different. This looks like it is here. This looks like it is. I do not know about the others, but many diseases are caused by point variants. So by the difference of one amino acid that can cause the disease. And here is just a set of diseases that are onset by variants, onset that are related directly to subcellular localization. So if you can cause a disease by subcellular localization, there's another indirect way of saying subcellular localization is one important aspect of function. Here is in fact a particular example where the mislocalization of this protein called PKC leads to cytokinases. Failure, and the way you see that somehow is that the microtubules grow in a way that is not normal. That's the control, and you see here they look different. And this is in fact, I believe I show you this image also to get an idea. This is typically what people see. This is where does the color come from? Do you remember that? So this is typically a tracer. There's, in this particular case, the blue and red fluorescent protein. Tracer means you essentially, you suspect that the protein, this particular protein here has some problem. You attach to it another protein. The green fluorescent protein was the first that people did. Now there's blue and red. And then they're always together. They're always together means that you somehow trace a compartment, you trace changes. Okay? But there are other reasons essentially why we want to break subcellular localization. It provides some information about the role of a protein. It does not say what the protein does. Knowing that the protein is nuclear implies there's a high probability that it binds DNA. It does not mean this protein binds DNA. Okay? So this is a very roundabout way of getting to function. But it is a roundabout way. In the drug discovery, simply, as I showed you, most protein targets for drug discovery are membrane protein, extracellular, cytoplasm. Here immediately we can just get rid of a bunch of targets if we want to do the same that we have been doing before. So again, it does not really allow me to say this protein is relevant, but it allows me, these 10,000s are not. That's the way it works. And again, this I said, some problems in it can lead to disease, and in fact it can help in validating protein-protein interactions. How do you believe this can be the case? How can the prediction of subcellular localization help to predict protein interactions? Again, it's a roundabout way. It's not a direct way. How could you imagine that?
I could exclude some protein-protein interactions by just seeing that they're in the...
Yes. Yes, if you have a protein that is extracellular and another one that is nuclear, if they met in a tubule outside of the cell, they may interact. But in the cell, they will not, because they will never meet. So in that sense, again, it does not tell you these two proteins interact, but it helps you to filter out proteins that do not interact. Let's get back to this idea of the zip code. And again, we do want to have a large-scale view of subcellular localization. And there are many experiments, but we still do not have the entire... this is for yeast. Yeast has about 6,000, well, about 7,000 proteins, roughly 6,600. And you see that about 5,000 of those have experimental evidence for their localization. Now, this is impressive on one hand. 6,600, 5,000, almost all. Right? But it's also true that yeast, so the first experimental study that determined subcellular localization for yeast happened 15 years ago. And that study was supposed to give a subcellular localization for every single protein in yeast. Every year there are many publications, and we still, so one way of looking at it is 5,000 and 6,000 or 600 is a high number. Another way of looking at it is there's still something open even after 15 years of very, very ardent research on this topic. Now, so we clearly need predictions on this issue. Again, there is sort of a pathway, a hierarchy in the way the transport is going through the cell, and there's sort of two different types of motifs. For the zip codes, the ones that are sequential, so you're just looking at a sequence, you will see them. The ones that are conformational or structural, such that you will only see the motif if things come together. This entire idea that there are zip codes, that in fact there's an address tag that makes proteins be transported, goes back to Günther Blobel. He was the first to phrase this idea in papers, and this is the 60s here. And in the 60s, this was quite a statement to argue that proteins would be transported through the cell by zip codes. He got his Nobel Prize for that in '99. And more recently, so in 2013, 14 years later, there were again 3 Nobel Prizes for details about the transport through the cell in the vesicle system. So Günther Blobel really got his Nobel Prize for the first idea for establishing the model and for doing many experiments. This is much more detail. How is the detail regulated in this transport? We can predict subcellular localization through the signals, through homology-based inference, through text-based analysis, de novo, and through hybrid approaches. And I'm going to go back today into, well, a mix. Actually, in some sense, it is a hybrid approach that mixes de novo and homology-based inference in a way that is slightly better. In order to predict subcellular localization, you always have to consider, and this is not subcellular localization. In biology, biological data is always a full stop. It's always what you have to remember. Okay? And that's important for the machine learning debate between neural network and SVM, and in particular for what I'm going to show you, which is sort of, in some sense, a surprising take on this. Prediction performance must be established properly. Now, this is a statement that you encounter in any machine learning, but in most machine learning environments, this is not as crucial as it is in biology. The reason, ultimately, is that most of the time that you do data mining, you do it for the sake of getting some data. Somebody's going to interpret this, and so there will be some action today, and the data mining you did will be over. So this will no longer matter. I'm sorry for that, I'd forgotten to switch it off. For tomorrow. Now, for the methods that we talk about, many of these methods are around for many years. So the lock tree, the one I'm going to present to you now, the original idea to lock tree is 10 years old, and has in fact produced results for experimentalists every day for 10 years. So you really should make a better effort if you really want them to be used to assess them. And the most important problem really is the one of similarity between training and testing. Again, this is the same for any kind of data mining. It is typically mattering less, because typically if your results live only for a week or a month, then you don't really worry too much about these things. In biology you have to. Sequence redundancy is in the public databases now. This is, again, I do not believe specific to biology. It's just something that you see. I believe in biology we have just a longer culture of 20 years of handling redundancy. This will be the same on the internet. I mean, just go to any Twitter account, look at the way people tweet. There's an immense redundancy in this. Somebody says A, and then you're going to see A 5 times. There's an echo. Prediction results must be interpretable for users. Now, this is a tricky one. And this is in particular tricky the more we move into, so currently in Munich we are debating heavily this issue of digital medicine. And in digital medicine, ultimately, we need machine learning for a physician. And physicians, when they have their meeting with you, have very little time. They cannot look at, well, this analysis says by SVM that something comes out. If I change the machine learning device or the Excel spreadsheet to another figure, then something else comes out. And the physician is not going to do that while he talks to you as a patient. There must be very simple conclusions that are understandable for the physician, where the logic of the system is understandable for the physician. In fact, there are a couple of examples that I know where people in fact developed a machine learning device to predict, in this particular case, the example I know is something for HIV. And the physician sees a rationale that has nothing to do with the machine learning device. So there's a rationale put on top that makes sense to the physician, because otherwise it would not be used. Okay, you may call this cheating, but ultimately what this is about is communication. Okay, that is an important issue. Benchmarking. So one of the issues really in the field is that benchmarking is very difficult, and it is difficult because the data generation is difficult. There's a lot of work that goes into it. And most machine learning applications, typically you get a data set that is 1 day. You sort out the way you do the machine learning, that is 6 months. You wrap it up, you publish or something, and about 8 months or something are gone. Okay? In biology, it's just completely the opposite. It takes 7 months to get the data, and then you do the machine learning device in 2 weeks, and then you rush through the publication. So there's much more weight on the data if you want to be survivable in some sense. Okay, let's get into what is log tree 2. Log tree 2, immediately I talked about log tree 1, that's the SVM that essentially uses as a model this pathway of sorting. Now one expansion of log tree 1 here is that now we go from, we include not only eukaryotic proteins but also bacteria. And here are a couple of other things that we want to do. We want to have more states. What I showed you so far was about 6 states. We have seen after the development of log tree that in fact people make sequencing mistakes. So a lot of the data that we use today comes from high throughput sequencing, and there are sequencing mistakes. Sequencing mistakes, one result of sequencing mistakes. So one is simply to say this is the wrong amino acid. Okay, the sequence is not A, C, D, E, but it's A, D, D, E or something like that. Another problem is that really the fragments are different. So rather than seeing that this is the protein that comes off the sequencer is this, that is sort of connected here. Or even as 2 different proteins, because it's not seen that it is one protein, it can happen too. Now, this implies that I have to have a different prediction method to tackle this. Remember, one of the main input features that we used to predict subcellular localization was amino acid composition. And what we did is we used the entire thing, right? In a case like that, if my input is fragments, this is deleterious, or can be really complicated. I also told you the story about NLS. I told you the story about signal peptides. Signal peptides, again, this is at the beginning, something very hydrophobic at the beginning. So as long as your fragment from the sequencing has the N-terminus, everything is cool for the signal peptide predictors. They essentially only use the first 50 to 80 residues. But if the first one would be cut off, that would be very deleterious to those. The NLS, as I told you, is a signal of many positive charges that can be anywhere in the sequence. That's relatively robust because, well, it can be anywhere, so the probability of hitting exactly that. Again, in those diseases, you saw it happens in real life. So it could also happen through a sequencing mistake. But that is not so dramatic. The problem is that most of the predictions are not based on errors. So yes, we can sort out, we can list nuclear localization signals, but most of the examples are N-terminal or other. So we need something else. We want to clearly put in membrane predictions because it's so important for drug development. And we want to have some decision tree. We clearly want to measure the reliability of it, and we want a high performance. So, as always, we begin with a data set, and we're going to build this upon some sort of SVM. So, with a data set, again, we take this. I'm sorry for the black. the background idea simply is this is a particular Swissprot record. In this Swissprot record somewhere there's a keyword that says subcellular localization membrane, nuclear membrane, right? So we need to identify the keywords. We need to distinguish this as an experimental annotation from other annotations that say by homology or by similarity or by blah, 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 blah. And we need to be able to recognize that this is in fact, although the word is, that is a nucleus and that is membrane at the same time. So and we need to, as a first issue really, come up and somehow filter the data. Filtering the data means, well, one way of doing is use the H-valve that I showed you before. Red things that don't have the same subcellular localization, blue that have the same subcellular localization. So once I have all of those experimentally known to know the subcellular localization, I take a unique subset. Unique subset means, oh wait a minute, I can keep it here. Unique subset, everything in that set is below that line, for instance, or some distance of that line. And then the numbers I get are roughly shown here. The total numbers, and this is redundancy reduced for the 3 different kingdoms, where the green one here are the eukaryotes. And you see, this is a tremendous loss of data. So this sounds great. You have 3,000, I believe this is, it's 3-4 years ago. But the ultimate thing that you can use here is much, much, much smaller. 35,000, not 3,000. Then you have to run through some stratified k-fold cross-validation, essentially training, cross-training, testing, and you repeat such that there is a split between training and testing set exactly in this ballpark, so there's no similarity between training and testing allowed. Shown here, so that's the annotation, that's the H-val 0, something like that, testing set, training set. Now comes the first issue. Somehow from the density that you see here, this is thinner, again, because you have redundant data, you throw out a lot of data. But it's also true, I don't have a lot of blue points up here in the training set. Is there any way in which I could increase the training data set?
Homology. If you allow homology, then...
Yes, you're right. I was too fast. Now, what you could imagine in your training set, you could pull in everything that is similar to that one as long as it's not similar to the testing set. Okay? So, yes, you add redundant information. And yes, that may not be very informative and it may not help you. But this is something to try out. Because remember the numbers, the bars, how much they differed from 35,000 to less than 5,000. So it's a dramatic step. This may really add important data. In this particular case, in fact, it did. It was very, very important for us that we used this idea. Now let's get back to the SVM. Again, the SVM finds the closest points and then finds a margin through these hyperplane through these closest points. It does that by projecting the problem into a larger space. If you want to find a similarity between 2 protein sequences, so similarity between 2 protein sequences, I believe I should have gone back another step. So now I want to have a prediction method that predicts subcellular localization. I do know that the signal is somewhere in the sequence, but I don't know where. I cannot run a sliding window because it just fails. We tried that and it doesn't work. Most likely because most of the windows in that particular case would not contain relevant information. I could try something like that, but I'm not going to go into this. So I want to have a method that addresses the issue of fragments. If somebody came with only this, and if that contains the NLS, well, if I know the NLS is trivial, but if that contained the NLS without me knowing the NLS, I want to find a method that also could find it. Somehow motif discovery or something like that. What could I use? So I cannot run a window. A sliding window, not trivially at least. But I do know, let's put it differently, and now we get back into the difference between machine learning and homology-based inference. So homology-based inference means that so A is sequence similar to B. B known experimentally. Then I can infer from the knowledge of B what A will be. Okay? Now, machine learning means that this similarity will be much lower, so that I can no longer infer this. But maybe what I have, machine learning essentially finds a path that says, okay, the similarity between A and a set of B, where B is a set of B's, many, many, many B's. None of these B's is enough to do homology inference. But as a data set, there is a signal that my machine learning has extracted that allows me to make the inference that in fact A has a particular localization that the machine learning device got from all of these B's. All of these B's could be even things that have different subset localization, right? This is essentially the way machine learning extracts information. But it implies that we ultimately still look at a sequence similarity. Okay, we need some measure of sequence similarity that can capture fragments or can predict based upon fragment, and also handles the situation that I don't know where the fragment in the protein is. So I somehow need to consider the entire sequence. What could I do?
Was hashing, but that doesn't make any sense.
What exactly do you mean by hashing?
Like the normal way you hash data usually, which means compress it somehow.
Yes. The question is the how, the sum. So the hashing, the solution, in some sense, okay, this is what I'm going to show you will be called a little bit of a stretch to the word hashing. But we did something that you may, without too much of a stretch, call somehow hashing. The question is, how is the detail to do?
Well, in this particular case, I would think of secondary structures, for example.
So you could follow all the secondary structures, and then what would you do?
Well, we definitely have something smaller to compare.
Okay, so we take segments of secondary structure, and for these segments of secondary structure, we have never tried that. So we haven't tried it because we haven't tried it. It may actually work. But I believe I would have discouraged you from trying that simply because my intuition is that it's not a single secondary structure segment. So a lot of information content is in something that people will refer to as helix turn helix. That means a helix, another helix, and something in between. That is very informative. In fact, those metal binding loops are always, many of them are helix turn helix. Some DNA binding parts are helix turn helix. It's like you grab the DNA with two fingers, or you hold the metal ball with two fingers. It looks more like this. While the DNA binding is really like that. So these motifs exist, but they are combining a couple of secondary structures. Now that makes it complicated because now you could look for something such as one helix, something, so three secondary structure segments. But then how do you move them? By always one? Maybe. But again, they are. So if you wanted to predict enzymatic activity, that I believe would be an interesting idea. And there is a sort of similar idea put into HMMs to characterize functional motifs that somehow is similar. But for subset localization, I still wouldn't do it. Any other idea?
Question, why can't we use sliding windows again?
Yeah.
Small windows, oligopeptides, I don't know. I mean, what we know is that in some cases,
okay.
For example, 5 peptides define.
It is true. It is true. It is true. For the NLS, absolutely true what you say. So, how would you propose to do it?
You just take every sequence of length of 5, for example.
Yeah.
And you use as an input for the similarity comparison.
Yep. Now we're getting somewhere. Okay. How would I do that?
So the motif idea.
It is a motif idea, yeah. It's actually, in some sense, if you want, so we tried the sliding windows in a different way. Now, have you seen the next slide? You were the only one who looked at it. So this is exactly the idea. Oops. String kernels. String subsequence. So this, in fact, is K-merged type of situation, where K is just the number. You said 5, so that would be K equals 5. So the string subsequence kernel, essentially they have three parameters, which is this K, and then a decay factor and this sort of gap factor that you have 5 mer, but one you may have an insertion between two proteins. This is the mismatch kernel. This is very similar idea. You say that you have a 5 mer, but you allow 1 or 2 mismatches, or whatever. You allow a certain number of mismatches, right? This is in fact the second free parameter here. Here's a profile kernel. Now the profile kernel, I'm going to explain to you, essentially now needs an alignment. So first step, you start with this multiple, what is typically referred to as a multiple alignment. So this is the sequence that for which you want to predict. You find related proteins, in this particular case, the homolog 1, 2, 3. And then you simply at every single position, so MKW here, the first 3 residues, and we go to the last. We count how often do you see M. What's the frequency of M? 1, so 100%, right? And K is 50%, and so forth and so forth. So on. You transfer that, the log frequency, into a matrix such as this. And then in this particular case here, what you do, again, this is the sequence, is a different one here. The 20 amino acids, that's the log frequency transferred matrix. Let's just begin with a K-mer length instead of 5,3 for simplicity here. And now you have to define a conservation threshold. So how conserved is it? So the way this now works, you build the protein sequence that consists of all possible 3 mers, 20 by the 3. Now you begin with the word cat here, and the word cat can, again, the mismatch idea can be different. It can lead to all of these. For all of these, you look up the conservation. If the conservation is above 5, then you say, this one I will not consider. All the others I will sum up, so that will make this 0 and all the other ones 1. You do that to the other protein, and now you simply do the vector product. The result of which here is 32. Okay? So this does something like the K-mer, but it considers profile similarities in some sense. So it in fact has the possibility through this counting simply to say we have positions that are conserved, that are the same through a bunch of proteins that I believe are related. I don't know their subset localization, but they are similar. And that tells me what is more important and what is less important. Okay? That's the entire idea. Method other than that is clear, right? Essentially you match exactly your idea, that you match two K-mers to each other. You look at the similarity between K-mers. Again, this example shows it for 3, you said 5, it's the same idea, same principle, except for now, in terms of counting the similarity, you consider the conservation. Could have done it differently. So our first approach to it was much simpler, where we simply looked at running windows that are conserved, and then we put them all in. This is much simpler. But this is much more involved. So this would find actually levels of similarity for things that are not conserved, if they're equally not conserved on both sides, in some sense. Could find similar patterns. Now, this slide here compares performance for some data set for different types of kernels. So the profile kernel is the one that I showed you before. Overall, that was a pre-test that we did, and that made us decide that the profile kernel does in fact the best, and that we go forward with that profile kernel. Now the next question is, how do we translate this binary classifier? So essentially what we have is a system that has 2 outputs. It's subset localization for its nuclear versus non-nuclear. And we discussed that last week already. How can we take a classification such as this and translate it into a multiple state classification? So again, the one against all idea simply is you do, if you have 10 classes, you do 10 different machines, and you go for the max or find some other way to go for it. You could go for an ensemble of nested dichotomies, ensemble of class balance dichotomies, of data balance dichotomies, and dichotomies, or a dichotomy with a fixed structure. This essentially means you decide how you build the hierarchy. This here means you build a bunch of hierarchies and try them all out at the same time, or actually average over all of those. There is no one structure. Okay? You have these 10 different things and you combine them in maybe 100 different ways. And you average over the ensemble of these 100 different ways in your final prediction. Class balanced is clear, so this nuclear localization would be essentially, we would postulate that nuclear and extracellular would be equally balanced. So the number data balanced is essentially now we put the balance in terms of the type, the way the data looks. This could be the same in some problems, and it could differ slightly. Okay, so we can compare all of this. The all against all is illustrated here. The nested dichotomies are shown here, so you have a bunch of these. I believe you understand that there are different ways of doing it. Let me just go back. In all of these dichotomies, in all of these hierarchies, in all of these ways, you begin with a system that starts at some point with a decision, and whatever follows is different. Okay? But all of these, in this particular case, use the profile kernel. That's what they have in common. Okay? So, in some sense now, we remember last time I said by this distinction, the first distinction would, in this particular case, for instance, be membrane or not membrane. By this, or secreted or not secreted, that's what we talked about last time. And by this distinction, the first one could have a different input than the subsequent ones. That is in this language here is no longer the case. In the profile kernel, we essentially compare two proteins based on their entire sequence. Oh, that's not quite right. In terms of informative k-mers, right? But we are not constraining the informative k-mers to any place in the sequence. We could do that. In this particular case, we don't do that. Initially, we actually did not do that because of technical reasons. So there are different classes. So archaea, we have the membrane, extracellular, cytoplasmic. That's all we distinguish here. Oh, here it is shown again. For the bacteria, we have a little bit additional. We have additional states. Fimbrium is the thing that makes a petal that hangs out of the bacterium, for instance. And for the eukaryotes, we have many more states. In this particular case, in fact, the first decision is membrane, not membrane. That's, in fact, the statement that I made. So the first decision is, do I go to the membrane? Membrane is everything here. If it goes in the first path here to the right, then essentially we talk about whatever is red here. If we went to the left, then, well, that's the whole everything other than membrane, right? This is the way the one minus the other one looks. It's just a visual presentation. And then we go further down here. Once we know in this particular case it is not in the membrane, so this red part that I showed before, the next part is secretory pathway or not, and so forth and so on. Here, what it essentially shows is that different dichotomies perform similar. Ultimately, we ended up with a fixed dichotomy simply because of CPU time. So if you build an ensemble of dichotomies, you essentially have to repeat the same thing hundreds of times. That was the only reason. How do we test? Well, again, so a couple of numbers to look at. Accuracy or precision, I talked about already. Coverage or recall is defined this way. Sensitivity. Now, one of the main things is standard error. Again, I put it up before, and somehow, if you take nothing out of my course except for this, then that is little, but it's something. So if you publish a method and that says 19.02, that's method A, and method B is 90.04. Can I have some comments on what is on the whiteboard? So the question to you will be, which one is better?
What are the values?
So that is, in this particular case, say an F1. F1 is typically defined as what I had here, accuracy times coverage times 2 divided by accuracy plus coverage. That's one way. Say that's what it is. So it's A better. So I want you to jump into my face and criticize what I put on the whiteboard. What's the first criticism? But ultimately the question to you will be, is A better or B better? Well, which method is better is actually?
So higher number is better.
No, you can't say that. I mean.
He did just say that.
Yeah, that's wrong. Sorry.
Good. Why? Why is it wrong?
Whenever you have one measure or only few measures of, you can't say which one is better because one might have an astonishing accuracy and the other might have
that is true.
Great coverage.
And that is a very, very good point. I'm glad you make it. But I'm going to force you to make a decision based on one number in this particular case. You're totally right. You're totally right. This is absolutely true, and this is the first thing to say. But assume since we have established this, let's ignore it for the time being. There's another problem here.
How much test data do you have to actually get this data?
1,000 proteins.
For both?
Yes. Oh, that's another very, very important thing. A very important question that you have to ask. Did they use the same data set? Yes. Do we compare apples with apples?
Then
we do.
The next question would be the third method, random one. 9.03 or 20.
See, random in this particular case is 10.11.
Very cool.
Yeah, very, very similar compared to random. This is, I'm really happy. Did we do this before? I can't remember. But you're asking me every single question that I would have wanted you to learn. So I believe I can retire. There's just one thing missing. And coincidentally, that's exactly what is already on the other slide. So the one thing that is very important, you have to jump up immediately when people give 2 digits. So one reason to jump up is simply the lazy effect. Because every single time you have to type a table with two numbers, you have to type 2 extra numbers. How many numbers do you have to type? And that ultimately is the question to you now. I believe what you said so far, I take as an argument they are equal because they're both so far from random and they look equal. But now my next question to you is, how many numbers after the digits do we have to put in to the table?
Well, according to the random, how many are significant, for example?
That's what I'm asking. Are you telling me or are you asking me? Or?
I'm saying, since the random is low,
yeah,
that's a rather significant difference.
That is certainly appears to be true. Here's the one thing that you never asked me about. You never asked me whether they are essentially the same protein, right? They could be. It could be, just for the sake of the argument, that's not what I'm after. But it could be, this is an important thing to learn, it could be that they are all extracellular. 1/10 of the proteins are extracellular, and essentially, or something like that, 90% of the data may be extracellular, essentially maybe one protein or 2 or 3 proteins, different proteins, and they simply say, whatever I see extracellular, I say extracellular, my data set is totally biased toward that class, so 90%. Random is still 10% because random does a proportional prediction. In 10 classes, say this would be my problem, right? This is the one bit that you didn't ask. But I'm still that is not what I'm after. And what I'm after is the question, what is significant? How do you define significant? And that brings us to this issue here, the issue of standard error. You have to absolutely find a standard error. Now, I give you a data set of, say, 1,000 proteins with known subcellular localization, or 10,000. How can you create a standard error out of it?
You check how many are wrongly classified.
Yes, and?
and then you build this, the standard error, you build a distribution graph.
No, but wait a minute, I'm not entirely sure. So tell me what I plot here. Accuracy,
Okay.
that is how many are correctly classified.
Okay.
Can anybody help him? Can anybody help me? I do need to plot, yeah.
I would say you have to perform the prediction way more often. So you use a k-fold cross-validation to increase the number of A, B, C's and whatever. So you get more output values and see how often you might not get wrong, but you are in which how often you get which F1. And then, yeah.
You're on the right track. Let's get back to that in a minute. You're absolutely on the right track. So assuming that my distribution, if I were to plot accuracy or coverage or F1, typically I would sort of expect a Gaussian-like distribution. Not quite Gaussian because we have 0, so there's a difference between 0 and 100 somehow, but it could be sort of Gaussian-like. For the Gaussian you need the average and the standard deviation. The standard error is essentially the number, but one is the standard deviation that you have here divided by the square root of the sample set. So square root in this particular case of 1,000. That's sort of, yes, you subtract 1 from that. So, but essentially that is sort of a rule of thumb idea. Now, since we have only one data set, we have to create a distribution somehow, artificially. One way in which we could do that is we could look at the differences between folds. So I do say a 10-fold cross-validation, that is what I believe I put on the 10-fold stratified or 5-fold stratified cross-validation. So then I would have in fact 5 different sets or 10 different sets. And I could look at the distribution, so it would not be such a smooth distribution, I would have essentially 5 or 10 points. That's true. But there's another way in which you can do that. In fact, this is much more precise and in many ways also simpler. Bootstrapping. You know what that is? So bootstrapping essentially is, so it comes from there's a famous German character, The Adventures of the Baron Munchausen, and the idea of this Munchausen is you pull yourself out with pulling your own bootstraps or your own hair. There are different ways in which he did it. Has been introduced by Bradley Efron in '79. And it's quite commonly used in bootstrapping, by the way. Here's an image of this guy who pulls himself, Munchausen, who pulls himself out along with his horse from the sump by pulling the hair. Right? This is an amazing guy, right? And essentially, while you may not entirely believe the truth of this image here in this story, it actually works for computers. The way it works is the following. You start with a set of predictions. Now you randomly draw from this set of prediction things out. So say you have 1,000 proteins, say you done your k-mer cross-validation. So essentially for every single protein you have a prediction. You label them, now you take out, say, 60%. Or there are different ways of playing this. The standard Efron, what most people do is they pick out a point, keep the information, put it back, and repeat that 1,000 times if your data set is 1,000 proteins. Okay? So putting back. You're not always sampling the same. In fact, it's very unlikely that you sample the same 1,000. So in biology, what we have learned is you get more conservative estimates when you pull out only a subset. Say 60%. This is something what we ballpark typically do. So we pull 60%, compile our performance, redo that. And now we redo that 1,000 times, which is very easy, very simple. No cross-validation involved. You essentially look up a table right? You randomize the table with 1,000 numbers, and you can even do that 100,000 times. This happens in no time on your little machine here. And then you get a wonderful distribution that in fact gives you a very good estimate of the standard deviation. What you ultimately measure is the consistency of the data, right? Which of course, now we're back to Munchausen. This is not really your error. But it is, well again, this pulling yourself out with a hair doesn't work. Even for me, it clearly doesn't work for you, but it wouldn't even work for me. But here, you at least get many of the problems that originate mistaken assessments of your own performance happen because the database is biased. The data set that you use is too consistent. And that you clearly capture here. Okay? So while you do not get the true, may not get the true distribution of how the method will behave for the data in 10 years from now, you clearly would see problems in the consistency of your data set today, or too consistent data sets, or to low consistency, or something like that. So you get a good estimate of that. Okay, and so far, in our experience, that has worked like a charm in most of these cases, that we now can use to assess this method. I already showed you that. Oops. For the first decision, and I admit that I should give you the random background. Here, at least I give you the error. The first decision here, whether a protein is a membrane protein or not, is made at 95 plus minus 2% by a specialist on membrane proteins. So that method, in fact, predicts membrane helices. And from the membrane helices, it says, well, if a protein has membrane helices, it's a membrane protein. Very simple. So now, we want to develop a method that predicts subcellular localization. We don't really want to predict membrane proteins. But the first step in our hierarchy happens to be this one. And it happens to be that one because in the fixed hierarchy that we chose, that made the most sense. And actually, it's also one of the best ones, if not the best one. So we have to compete with this one. Otherwise, we could just throw our SVM away and use the membrane prediction method. And the number is missing here. I'm not sure why. And then we go further down in the hierarchy and get lower levels of performance accuracy. And you see in many of these cases here, the error shoots up tremendously. So here is an assessment, is a value of 38% accuracy with a plus minus 48. This is, of course, I'm not entirely sure how to call it. You could argue this is nonsense. As long as you, however, quote your error, it's very clear for everybody to see how much you can trust your estimate. So clearly you cannot trust it very much. But it's also true that you have a classifier there. All the data points that you have, you classify in some sense, and you have to report a number for that, right? And if anybody reported a number of this type here, well, if anybody in a paper, and you see that again and again actually, not only from students, but from real researchers, that people quote maybe another digit here, many levels of digits, and then you look at the data and you see something like plus minus 10. A total nonsense, right? By the way, if it were plus minus 10, how many digits are supported? 3?
No, 2.
So,
All after the dot is not important, because 10 is
plus minus 1. What if it were plus minus 1?
plus minus 1 is that.
No, I'm just asking. So I removed all these digits. So essentially, I'm asking you, if I had a standard error of plus minus 1, would that support anything other than integers?
I don't think so.
No, clearly not. So the real value is already gone with a plus minus 1, right? With a plus minus 10, well, now you can argue, okay, you know, there's essentially, I cannot distinguish between sorry, I cannot distinguish between 28 and 48. So that's another way in which you could quote it as a range. The problem with that is simply the readability. But it's the truth, right? As long as you give the truth right, and as you describe it right, there may still be a signal in there. This may still be relevant. It may be that this, in fact, is not a problem of the method. It's just a problem of the data set. You just do not have enough data. Somebody comes with new data over the next 10 years, and the method may still work very well, or may work, well, 38% we may call not very well, but then it's also after many levels of detail here. Okay, so this is a bunch of different numbers. This again is making the same mistake that you acknowledged before, Andre, which is to in fact have only one number, but it's an overall number, and this simplifies some comparisons. So the number behind here, the Q, that's the quotient, Q5 means this is a 5-state prediction. Okay? In the 5-state prediction, we have two methods that we compare, and in fact the ones that I put on that Tanya put here, we looked at a bunch of methods that performed that are somehow competitive, or the best competitors out there, and for every single pair here we picked the next best method. Let's begin here. This method that I showed you gets that level of a 5-state accuracy for bacteria, and the best competitor out there is this one. Is log3.2 better?
You can still, considering the error, go again for both at 90%. I don't know, this is accuracy? Or?
No, in this particular case it's really, yes, it's something else. So it's the 5-state accuracy. So meaning you look at all 5 classes and you sum in all the 5 classes how many of the classifications are right. So in this particular case, coverage and accuracy is the same.
Okay.
Make a prediction for every single case.
It is better. Log3.2.
But not significant.
Not significant.
No. Absolutely not. This is after a long development here, you get something like this, 86 versus 71, and you are very happy. But then you look at the error rate and you have to admit, okay, the value is higher. Okay, I'm happy that it's this way. I'm happy to be the developer of log3.2 and not of psort. But I cannot state that my method is significantly better. Maybe if I have a... So there, in fact, in our publication of this, we argued all kinds of other things. There are other reasons why we believe we do well, but at least this is not established, right? In this particular case, it's the same thing. Here, we compared it to our own method, so the runner-up here was our own method. And here, again, it's the same story. So for none of these data sets could we establish... by the way, I have to say something here about the data sets. What we did here is we developed the method, and then we looked at proteins that were added experimentally since we had developed the method, so that nobody had used before. So and then there is a clustering of this data set, so that's why the data set is very small, that's why the high error rates are so high. Okay, here's another way of looking at it. Another way of looking at it, we wanted to, in fact, address the issue that people come with fragments. So what we're going to do here is, this is the full length, and these are in fact, now this is a larger data set here. And in this larger data set, now it is clear that log3.2 is higher. It is significantly higher, although not impressively high, right? We are just essentially one standard error away. And now we take away the N-terminal signal. And you see two things. First, this one goes down significantly. So many of the k-mers obviously come from the N-terminal signal. That's clear from this. It's also true when you look at how much this one goes down or that one goes down, that is much more so for other methods. This one here, we remove the C-terminus, and again, there is some signal for some of these methods, but this is small, and it's also small for us. we randomly remove 1/3 of the protein, and you see now again, this is similar to removing the beginning of the protein. You simply, by randomly removing 1/3, very often hit into the motifs and the k-mers that are relevant. I'm sorry. And again, this is the same here. For these methods, what you conclude is that they really explicitly use the signal peptide. So that's why the reduction from here to here is much higher than from here to here. Same thing here. For us, it's sort of similar because we have a method that uses the k-mer spread all over the place. But ultimately, at the end of the day, we believe, at least for a variety of ways in which you can make mistakes, not only do we do better at the beginning, but also for these mistakes, this log tree 2 does better. Now, there's another step, log tree 3. Log tree 3 simply puts homology-based inference into the equation. And the simplest way in which it does it is it says whenever we can establish homology, we take the homology hit, the homology lookup. So again, I know another protein for which I know the subcellular localization, I take that. Whenever the psi-blast value is above a certain level, and otherwise I take the SVM profile kernel. And that shows the performance for a bunch of different classes, not for all because it's 18 different classes, for 1600 eukaryotic proteins. The performance of the homology-based inference is blue, the performance of the de novo is red, and the performance of the sum over the two, or take this and that, is the green, and the green is always for all classes above, or at least as high as the best of both. So finds a perfectly good path. Overall, this is the number of classifications. Then that leads to an issue where we have to come up with a reliability index. So a measure for how reliable my prediction is at any single point. That now becomes an issue. In the SVM, it's a probability. It's a lookup. It's relatively easy to do that. For the homology-based inference, it's not that easy. So we have to sort of find a way to normalize our score, and then at the end of the day, we come up with a prediction that we can then use to look at entire organisms. So when we look at human, that's plasma membrane, that is the part that is extracellular, that is cytoplasmic, that's nuclear. And now we get back to the issue I said at the onset of my lecture. Most drug targets are against membrane and extracellular proteins, so this in fact reduces the number of potential drug targets quite a bit. So essentially, if I wanted to look only at membrane, is this fraction, extracellular membrane is this fraction, but that covers already in the ballpark of 70% of all my drug targets, and it clearly throws out more than 70% of my human proteins to look at. You immediately see how functional, how helpful such a prediction can be. You also see that for different organisms, this is orangutan, just to put the point how surprisingly different they are. We don't quite know why. But overall, you immediately see that nuclear proteins do change. Again, this is over a wide variety of eukaryotic proteins. There's only those two that are mammals here: fish, plant, chicken, yeast, single cell eukaryote. But you see there's quite a difference. You could also look at the difference. Now, I do not remember which one. I believe the inner circle is the difference between, so this essentially is the difference between the ones that we know experimentally and the ones that so if we only looked at homology-based inference, so we only looked at the ones that are experimentally characterized, then this would be the pi, since we now can predict for every single one. So for yeast, remember I said there's 6,600, 5,000 of those have experimental information, that's the inner pi. And this is simply the prediction for the 6,600. And for some of those, you see, in particular here, you see important differences. For others, even here for the nuclear ones. So there are important differences. So the prediction carries a very important different way of looking at it. So the differences here, since our method is fairly accurate, the differences here are not really the differences between prediction and experiment. The differences are between a subset of all the proteins for which you have experiments, and an entire set for which you have predictions. And it matters in this particular case. Okay, so this concludes our part on localization. Thursday there's no lecture because of the Dies Academicus. So next week we begin to get into protein interactions, and then next Tuesday, on the 15th, is Jana Schmidt, data mining in healthcare. Thanks for your attention.